Hi, this is Dr. Jibeli, and in this video, we're going to talk about the written answers for the topic 2.3, which is all about carbs and lipids. Remind you that all these questions are pulled from previous IB exams. So we're going to look in how the questions are stated and try to translate some of those words. And at the same time, we're going to learn how to be able to answer them. So the first question here is a nomogram. The nomogram is all about BMI and looking at the height as well as the weight of a person and trying to figure out if they are obese or clinically obese, overweight or underweight or in a normal weight. So here is the question. It says using the nomogram, state the lower weight limit. So the lower limit, that means the minimum of a woman with a height of 155 centimeters who is classified as overweight, giving the units. Now, here's the important thing. They ask you to give the units, but I'm already telling you from now, every time there's graphs and you have to pick up things, make sure to put the units because they always ask for the units. So even like right here, they did mention the word give the units, but sometimes they don't say that, but they do require you to put in the mark scheme the units. And they will do it as a trick. So it's important, if they ask or they don't ask, always put the unit. So first the question says that her height is 155. So that means in the height here, on the bottom on the X, she's right here in this little dot red. Now the green. The next thing is that they says it's in the overweight classified. So that means if I highlight the overweight here, this is the section where it's overweight. So as you can see, the blue highlighted, that's the section that they are talking about. Now we have to find a line in which the 155 weight will go. So it's straight line here and it hits right at this point right here. This is the minimum weight in which this woman will be considered as an overweight. But of course, we have to look at the y-axis to see what's the number. If we go to the y-axis, you can see that it's 60. So the answer is 60. But it says give units. So the unit, you can look here that it's written kg. So sometimes the units will be in the graph. Just make sure to look at these units and put them in your answers. It's very straightforward. You don't have to do out of the blue. So the answer would have been 60. As you can see, it's 60. It's a very easy graph to look and analyze. And it's basically something that you have to learn from 2.3. Now, another question that can come out from this is that says here, state the major health problem of the circulatory system. When I say circulatory system, it means the heart that is related to obesity. In this case, it can be thrombosis, it can be stroke, right? It can be coronary heart disease. And when I say the word coronary heart disease, the word the coronary is basically a vein that it's a blood vessel that is found in the heart, in which supplies the heart with blood. So if this guy gets blocked, then most likely the heart is not going to be able to function well. So let's look at some examples of the options they gave. So we do have coronary heart disease. We also have thrombosis, we have stroke, hypertension, even arteriosclerosis, which is one of the information that you have to know as an application in 2.3. So you can mention any of those ones and you would definitely get the points very quickly. Now let's go to the next question. This question says draw. So when we draw, remember that draw is not just drawing. Make sure you always also labeling because that makes a big deal. Now it says the structure of fatty acid. Now the structure of a fatty acid in this case, it's basically a chain of carbons, and you can see here. Now how many carbons doesn't matter. Try to draw between the 7 to 9 or 7 to 10. We have here 6, 7. Next you have to basically up with the hydrogens. We have to fill up because remember carbon can do 4 bonds. So you have to attach them to 4 things. And you can see all of them is attached to 4. Right? There's 4 bonds surrounding the carbon. This one, we have 1, so we need to have 1, 2, 3, and 4. That will complete. But that's not the whole fatty acid. You still need to be connecting it to a carbon that is connected to an oxygen and to OH. Now, this is a structure of a fatty acid. Now, you can always label, now this is saying the hydrogen, the carbons, the single bonds. But this is definitely the structure that they're looking for. As you can see it here, it shows basically the hydrogens with the carbons, right? And then here have the carbon with CO. So basically, what are they asking? They want you to do the hydrocarbon chain of single bonds, okay? Because it said the word saturated. Saturated, that means there is no double bond. And it says at least 4 carbons. Now we have the CO head, which is this right here, and the 3 hydrogens on the other hand. So they want to see that. They want to see the 2 edges. So again, the minimum of 4 carbons. So I usually put 7 to 8 because that's what is more commonly used. But go ahead, this will be an example of a structure of a fatty acid. Now let's move to the next question. The question says distinguish. And when I say the word distinguish, it means give me the difference. And again, structure of a different types of fatty acids in the food. Now we have different types of structures. We have the saturated. We have the unsaturated. And we have the polyunsaturated. Now, what's the difference between them? Saturated, that means, remember those chains of carbons? It's just a single bond. That's it. It's just single. Now, unsaturated will mean that there is a double bond going on, at least one. So in this case is one double bond. And usually they put mono here. If you put mono unsaturated, it clearly says that it's one. Now, when it says the word poly, poly means many. So in this case, you have a double bond here, maybe another double bond here. So you have 2 or more double bonds. So that's talking about their structures. All of them has the carbons. Of course, I'm not drawing all those hydrogens here. You could do that, but I'm just focusing on the singles and the double bonds. We also know that saturated in this case, the saturated is the bad fat compared to the unsaturated, saturated, which are the good fat. But this is how you would distinguish between them. Now let's look at the mark scheme. So the first one says fatty acids can be saturated, unsaturated. That's what we mentioned. Second, unsaturated can be mono or poly, as I have mentioned. Third, saturated have no double bonds or maximum number of hydrogens, which you can see, while unsaturated have at least one double bond, or the poly has many double bonds. So it's basically all the drawing that I have done. You can do the drawing or you can put them in words. Now, here is an additional thing. We have cis bonds found in hydrogens as well as trans. What does the cis and trans mean? So cis and trans is that you have these carbons with double bonds. The hydrogens could be located on the same side, and that would be known as cis. And then if the hydrogens are in the opposite sides, that is known as trans. Now, trans is the worst kind of fat. Cis is a good fat. So here you see also trans has hydrogens on the opposite side of the double bond. And then the last thing is the length. The length of the hydrocarbons, that means that chain of like this, it varies between different types of fatty acids. Also, the double bond, is it here? Oh no, maybe it's here. Oh, maybe it's there. You don't know if it's in the first or the second or third carbon and so forth. Now, of all of this, the most important thing is that they do accept a diagram to illustrate these marking points. So all that drawing that I did here in the beginning, let's go back. They accept these drawings here that I did right here. But besides this, I would have just add the cis and the trans by showing the hydrogens here, and then the hydrogens on the same side, which is cis. Of course, I have to label them, right, trans. So from me just drawing all of this, you're already getting those points. So remember to always draw, because drawing is a huge plus in IB. And even if it doesn't say draw it on the sides, because I know some of you guys have a hard time writing. So sometimes drawing and putting little labels next to the drawing can get you points, okay? Now let's go to the next question. This one we just answered, which is compare and contrast, right? Which is give me the similarities as well as the differences. So compare here is similar and contrast means differences. Of who? Cis and trans. So both of them, as you can see, they're both made of double bonds, right? And both of them, they are also what it calls, they're part of the unsaturated fatty acids. But the difference is that the cis have their hydrogens on the same side, while the trans have them in the opposite side. So these are basically looking at their similarities and contrast. So they can see both are unsaturated and they both have a double bond. So this is the case you can see. A lot of students, oh, they think it's 2 points. Be careful. This is just 1 point because it says or. So when you put those 2 points, you already think, oh, I got enough. You got to be very careful. The more information you have, the more you write. Next, the cis trans have 2 hydrogens on the same side, while the trans fatty acids on the opposite side. That already gives you the second point. Now, as I mentioned, cis are more healthier than trans fatty acids. Now, look, all of this is still considered as 1 point. That's why IB can be such a tough situation, because sometimes you're thinking, oh, I wrote these 2 things, those are my 2 points. No, sometimes they're considered as just 1 point. That's why the more information you have about something, the better it is. You also know that cis has a lower melting point compared to the trans, and of course cis has a kick, which kink means more like a bending going on compared to trans. And here, accept annotated grammar. Again, drawing is very important. Now let's go to the next question. Next question says here outline. So basically it's talking general. Remember that you can also draw. Now the role of condensation and hydrolysis of who? Of fatty acids, glycerol, and triglyceride. First, let's remember that condensation, right? Condensation is basically building. So who are you building here? You're building molecules. In this case, we're building triglyceride. Now, how are we building triglyceride? Because triglyceride is a glycerol plus 3 fatty acids. Now, when you're going to build them together and join them together, what's going to be released? It's going to release water. So every time you build something, you release water. Hydrolysis, on the other hand, is the opposite. This one is breaking. So when you want to break a triglyceride, right? You're going to break them again to the 2 pieces, right? You're going to break them to the glycerol and the 3 fatty acids, right? So you're going to break them down. But to do this, you're not releasing water. This time you add water. So you have to add water to break them. Now, how much water would you add? You're obviously going to add 3 water because there's 3 fatty acids. See, you have a glycerol attached to 3 fatty acids. So what do you really need here is that you need a water to break this one, a water to break this one, a water to break. So do need 3 water to break those fatty acids. So that's the difference between condensation and hydrolysis of a triglyceride with its fatty acids. So let's look at the mark scheme. You can see the condensation reaction here occurs between 3 hydroxyl groups of glycerol and carboxyl groups of 3 fatty acids. Now here's in more detail, but IB does accept if you just mention condensation occurs between a glycerol and 3 fatty acids. So just saying glycerol and 3 fatty acids, you will get those points. Next, it forms triglycerides, as we mentioned, and of course you can mention the 3 waters that are being released. You also can say the bond between the glycerol fatty acid is ester linkage. You are not required to know it, but it is good to know because sometimes mark schemes can add extra information and it will help you. Now, what is ester linkage? That's the bond between fats. Just like peptide is the bond for proteins and glycosylated linkage is for carbs. So the more you know the bonds for the different things, for carbs, proteins, and fats, the better it is. Now, when one fatty acid is replaced by a phosphate group, phospholipid is formed. Now, this is the killer because nowhere in here did they mention phospholipid. They mentioned about triglycerides. So it's like, how would I have known? That's why I always tell students, sometimes if you know more information about other fats, add them. Add them. You're not gonna lose points for adding more information. You're only gonna lose points if you contradict yourself. So here, you're just saying that glycerol, right, is added by 3 fatty acids. But if I take one of, oops, this is too big. Let me go back. If I take one of the fatty acids here, right, and I put a phosphate, this right here literally turn into a phospholipid, which is also a fat, but this one, phospholipid, is the one that you find it in the cell membrane. So it's just saying you're switching out triglyceride and putting a phosphate instead of fatty acid. Who would have known that this would get you points on this question? What happens is sometimes IB adds more things and you're like, I would have never known this. That's why getting exposed to previous IB exams and mark schemes can give you a little bit of an enlightenment for more information. And then when we talk about hydrolysis reaction, here is the presence of water and they break molecules down into their subunits. So right here, you can see there is glycerol And then you have the 3 fatty acids. And then this is basically showing a condensation where you can see here, you form the triglyceride and you release 3 water. So you can also have the drawing, but it's not required to draw because it just says outline. But I usually tell students to draw. If you know how to draw it because you can get the points in there. Now let's move to the next question. The next question says, here's state. State means just listing and 3 functions of lipids. So we just need to mention the 3. Usually when it comes to state, the first 3 things you mention are what they're going to pick on. So you cannot just go and write like 15 things and expect that they're going to look at the 15. No, they're going to look at the first 3 only, the first 3. After that, they stop looking at whatever answers you have. So make sure your first 3 are your best answers first. So when it comes to lipids, what are the functions, the main functions that we know? One of them is insulation. The other one is energy storage. Another one is protection. And there's so many other ones that we can talk about. Buoyancy. So let's look what they have here. First, phospholipids are the main components. So structure-wise, look what they mentioned here. Phospholipids are the main components of cell membrane. Have I thought about this here? No, I only mentioned these. So you can also mention a structure. And he gave examples, such as phospholipids. Next, hormone. Hormone, steroids. Steroids are hormone, like testosterone, progesterone. Did I mention it here? No, but it's hormone. Would you get those points off? Not really, but here's a point about IB. Sometimes IB will be very picky because they're going to say, no, we just want these. But as an IB examiner, if I saw that you wrote insulation, energy storage, protection, and if it's not on the mark schemes, we can kind of write up for IB and say, hey, these answers are legit, so that means you should add to the mark schemes. And sometimes they do. So moving on, we also have insulation. That's one that I mentioned. Next one, we have protection, as I mentioned. Now, do you have to write all these stuff? No, because it only said state. So only if it says state and give an example. So if you just mention these guys without the extra information, you still will get the point. So just structure, hormone signaling, insulation, protection, and storage energy, as I was mentioned. So I mentioned these 3. So you still get the points. But like I said, don't panic. Even if you write something that it's logistic, and we know that it is, we can always go back to IB and say, hey, this answer is also legit. And sometimes they do change it for the consideration, especially if a bunch of students have written something. The exact same thing. So that will be some of the functions of lipid. Moving on to the next question. Here it says compare and contrast. Again, the word compare and contrast means the similarities and the differences. And that means we're going to look at the differences and similarities of both lipids and carbs as energy storage. Now, a little scoop to remind you about this. So we know that carbs, these are basically short-term energy. What does that mean? That means the energy is used quick. And these go for like high-intensity exercises. While lipids, on the other hand, this is a long-term energy. And this is more for the slow ones, like what if you go for, so this is like going for a run and this is going for a walk, right? Now, carbs and lipids, if it's a complex carb, right? A complex carb plus the lipid, this similarity between them that they're both insoluble in water. Again, I said complex carbs, not all carbs, because if we say simple carbs like the small subunits like disaccharides, the small pieces of sugar, they are soluble. When it's a big, huge carb, they're not soluble in water. So they both basically are examples of storage of energy, right? Both are examples of storage of energy, and they both are in soluble water. However, the differences between them is that carbs are usually short-term and lipids are long-term. Now, carbs, they are stored basically as glycogen in our liver as well as our muscle, while lipids are stored as fat deposits, and there's a specific name for that, which is the adipose tissue. So these are kind of differences that I'm just putting out there that I know of. Now, let's look at the mark scheme. Here it says for the similarities, complex carbs and lipids contain low chemical energy. It can be used as energy storage. So you just mentioned this, that the both of them are energy storage. You got the point. Second, complex and lipids are both in soluble water. As I mentioned, they cannot be easily transported. So those are the two similarities. Now for differences, we have lipids molecule contain more energy per gram than carbs, twice as much. That is true because for lipids it's 9 times, while for carbs it's 4 times. So let's say if you have 1 gram of lipid and 1 gram of carbs, what would you see is that that would be giving you 9 calories for fat, and that would be 4 calories for carbs. So you can see that the lipids contain more energy per gram. Another one is carbs are more easily digested than lipids, so they release energy more quickly, and that is true. That's what I wrote. It's a very short time. It's a very quick energy. Another difference, monosaccharides are water soluble and easier to transport than lipids. It's true. So not the complex, like I said, the monosaccharides. That means that 1 piece of unit of glucose or 2 pieces of them, they're small, so they can easily be going through the water and the blood. They're easily transported. And they're much easier transported than lipids too. Animals tend to use carbs primarily as a short-term energy, while the lipids are more like a long-term energy. That's the most common thing that students know in IB exams. And carbs are stored as glycogen in animals, and lipids are stored as fat. So you can see here, again, some of the things that I mentioned. Now, depending on how much points this thing, if this is 4 points, right? If they ask you for 4 points, usually I will put 2 similarities and 2 differences. That's what I hit. If you don't remember, at least make sure you have 1. Because if you have 4 points and you only mention 4 differences and you don't mention any similarities, you're not going to get 4 points. You're probably going to get 3. So make sure that you have half, or at least you have 1 for the other. So you can be like 1 for 3 or 2 for 2. Remember to always balance your answers with both sides. Now, the next question, list 3 examples. Now, list means just put the names in there for monosaccharides, disaccharides, and poly. So when I say mono, in this case, we're talking about 1 unit of what? Saccharide means sugar. So when it comes to disaccharide, to monosaccharide, we're talking about glucose. Glucose is the most known one, right? We also have galactose, and we also have fructose. So these 3 are the mono. When we talk about disaccharide here, disaccharide means 2 units of sugar. So we have, for example, maltose, we have sucrose, and we have lactose. Again, they only list 3 examples of each, right? So I did 1, 2, 3, and then I have 1, 2, and 3. And now polysaccharide, these is many units. And this ones we can put cellulose, we can put glycogen, we can put starch, and there is chitin. So there's many other ones out there. So these are the 3 that I put for polysaccharides. Now be very careful. It didn't say 1 example for each. It said list 3 examples each of. That means 3 for mono, 3 for di, 3 for poly. So let's look at the senses. Mono, as we said, glucose, galactose, fructose. For disaccharides, lactose, maltose, sucrose. And for poly, we have cellulose, glycogen, and starch. Just as I mentioned. If you remember these really well, then you have easily answered the question. Next, describe. So this basically describe, we're talking about writing in words, details, like you basically telling the details of the structure of the starch. But in this case, it's not just the structure, it's also the function of starch in plants. So that means putting in words, what does the starch look like and what does the starch do? Now, especially in plants. So first of all, we know that starch is like a storage in plants. We also know that starch has 2 types of structures, right? It has amylose and amylopectin. We also know that starch is made of glucose plus glucose plus glucose. You know, it's just a list of glucose going on, which is the same thing as saying is made of maltose plus another maltose, because maltose is made of glucose plus glucose, and it just keeps adding more and more and more, right? We also know that Starch can also be used as energy by the plant, right, as a storage energy. So what else could we mention here? We have mentioned that it's a storage and gives energy for the plant. That's as a function. As a structure, we have mentioned that it's made of large amounts of glucose, basically maltose, and it has two types, amylose and amylopectin. So I kind of gave some of the information. They can give also the structures, what this is 1 to 4 linkage, and this is 1 to 4 plus 1 through 6 linkage. That's the structure of these guys. So let's look at the mark scheme and see what they really are asking for. So when it comes to structure, it's a polysaccharide composed of glucose molecules. That's exactly what was here, right? Polysaccharide made of glucose molecules. Contains amylose, which is linear. Now amylose, as I said, 1,4 linkage is linear. What else do we have here? Contains amylactin, which is branched. So the linear means 1 through 4, and branch means 1 through 4 plus 1 through 6. So as an IB, I still will give you the points because you have shown that there's different linkage, which is branched. So if you don't remember these numbers, you can just say linear and branched molecules. Now functions, storage in plants. And the storage form does not draw water. So this is not necessarily you need to know about drawing the water, but this main one, the glucose energy in plants as a function, it gives quick energy. That is considered your points. Next question, describe the structure of starch, which we just explained right now, right? So it's plus glucose, plus glucose, plus glucose, and it keeps going on. So it's a polysaccharide full of glucose. So it's a starch, is a carbohydrates. Now this is the most obvious things that we usually lose the points is because it's the obvious. We're ready. Oh, we know that starch is carb, but we don't think of writing that in the test. That's why put the most obvious answer. We know that starch is carb, so just by mentioning that it's carb, you can get the points. It's formed of carbon, hydrogen, and oxygen. That's another thing that students don't put it up. So when you see describe the structure of starch and you see 5 points, that means they want as many information. So you know that first it's carb. Then think about how it's made of these hydrogen and oxygen. It's using these 3 elements. It's a polymer, it's a polysaccharide. That's another thing that we didn't think about. Next, it's formed from monosaccharide simple glucose. So you can say it's made of glucose, it is a polysaccharide, it's a carb, and it has these elements. It's linked together by condensation. Who would have thought that this will be in the mark scheme saying what is it formed from? So when you talk about the structure, we're saying, oh, it has these units, right? But the question is, how did you put these units together? Oh, through condensation. So here is just open your mind. What are the possible things talking about it? So you talk about where they come from, which elements they have. Is it a poly or is it mono? And what kind of sugar you're using is glucose, and how'd you put them together through condensation. And of course, different examples of structures, which is amylose and pectin, giving examples. And then you mention about who they are. Like amylose is unbranched, which is linear, and while amylopectin is branched. So here you're trying to pull out as many information about starch. So just before I can go to the next one, just the more you look at these mark schemes, the more you'll get information of things that you could write about this. Because a lot of students will see the word describe structure starch, and they obviously would just say it's made of a bunch of glucose, and they're like, what else could I possibly put? Well, put the obvious. They're carbs. Talk about the 2 examples, amylopectin and amylose. Talk that they have hydrogen, oxygens, and carbons. They're like, really? Yes, the more information you can put, the most obvious information, put it down because you can get points for it, okay? Moving on to the next question. Here says distinguish, again, which means the difference between amylose and amylopectin. This is something that we have been repeating it. So amylose, we know that it's linear, right? While amylopectin is branched. That should be giving enough information. You can put the 1 through 4, and this is 1 through 4 plus 1 through 6 linkage. And of course, you can say one is more digestible than the other, such as amylopectin more digestible than amylose, and that's pretty much it. But here it says unbranched for amylose and branched for that. So this is usually probably a 1 answer, so that's 1 point. Just mention linear and branch for each one of them, and you're good. Next question says, outline how the structure of cellulose makes it suitable as a component of cell walls. So now we're talking about the structure of cellulose. So think what the cellulose is made. Well, first we know the cellulose is made of glucose, right? It's a chain of glucose. So that means it's polysaccharide. Okay, it can mention that it's a carb. It can mention the glucose, that the chain of glucose here is basically made of these chains of glucose that are very strict with each other, and it's alternating. So the glucose, what I mean by alternating is if I draw here, oops, let me write this. This is the glucose, and it has the chain here, it's alternating. The next one would have been completely opposite. It will be down here. So it's just the alternating of the glucose structure levels. And of course, you can say the cellulose is not digested by us. Why is it not digested by us? Because we do not have the enzyme to break cellulose. We don't have cellulase. So let's look at examples. It has a chain of 1,4-glycosidic linkage. Now, that's because, again, as if I look back here, there is the 1 connecting to the 4 of the carbon. So carbon 1 is connecting to the carbon 4. So that means this is linear, and you can see that it's linear. Now, glycosidic linkage is basically the bond between them. As you can see, I draw here, I can draw a bigger one showing the straight line. It is beta, of course. Well, this is one of the reasons why we don't break, because we can break the alphas, but we cannot break the beta glucose. Okay? So you can say a form of straight chains, form microfibrils and longer 10 microfibrils, parallel bundles of cellulose molecules, hydrogen bond, the cross language between cellulose molecules hold them together. Now this here is more details, and of course many students will never know about this. So the only thing you need to know is they forms microfibrils, which is basically makes the structures of the walls much stronger. Then we have high tensile strength, which is rigid. So if you just say it's rigid and it doesn't stretch, so provide support, allow turgidity, which means turgidity means keeping the plant standing strong and tall and it's not bending. So just saying the word rigid, it's good enough. If it has a lot of, so cellulose makes the cell wall so rigid and strong so it doesn't bend. And that's pretty much for the questions when it comes to 2.3. So looking back, these have future questions that could fall, such as why do humans have glycogen instead of cellulose? It's basically this one, right? Because again, the beta, we don't have the enzyme to break the beta, we have the enzyme to break the alpha glucose. And we look at different types of lipids and the different types of healthy and unhealthy fat. So these kind of examples, you can also go back to my lecture on 2.3 and you can answer these questions. But these are probably the future questions that can fall on IB exams. So 2.3, again, try to remember the structures of cellulose and of starch, because these guys, starch and cellulose comes a lot in IB exams. Also be able to give different examples of fat and know the nomograms. So these are the most common questions that comes from 2.3. I hope it clarifies or open a little bit more ideas of how to know the information for 2.3. And that's pretty much. Welcome.